Hello everybody, I am Giorgia Biasini, PhD student from Sapienza University of Rome, working at Antidoping Laboratory of Rome. Thank you for the opportunity to describe my research project about the detection of altered markers of red blood cell storage and its potential application for the development of a direct method for the detection of autologous blood transfusion in doping control. One of the greatest challenges for anti-doping authorities is to detect the illicit recourse to autologous blood transfusion for doping purposes. This prohibited method is still used by athletes to enhance their physical performance by increasing the oxygen delivery to muscles. It consists in the withdrawal of a certain volume of blood, its storage for a sufficient period to allow the reinstatement of the original blood volume, and its reinfusion. Currently, the approved method for the detection of autologous blood transfusion is the longitudinal monitoring of specific blood parameters of the athlete's biological passport. This indirect method of detection has some limitations, such as the undetectability of microdosage transfusions and the generation of confounding profiles due to particular physiological conditions. For the above-mentioned reason, there is the need to develop a direct method for the detection of autologous blood transfusions. The aim of the present project is to set up a pool of direct biomarkers whose level change over storage time and can still be traceable in reinfused sample. The target proteins of this study are BAM3 protein, the major red blood cell intermembrane protein with a key role in maintaining cytoskeletal organization and membrane integrity, peroxide redoxin 2, the main antioxidant enzyme of erythrocytes, and glycophorin A, the major sialoglycoprotein of red blood cells membrane. To this goal, we analyze level of these proteins in blood samples at 3 different time points, immediately after the collection, after 20 and 40 days of storage, under standard blood banking conditions. Protein levels were analyzed through SDS-PAGE, Western immunoblotting assay, and by flow cytometry. Moreover, we isolated red blood cell fractions through a Perkle gradient depending on their density that correlates with age. Peroxyredoxin 2 and BAM3 levels of each fractions were analyzed via Western immunoblotting assay. Finally, we evaluated the suitability of dried blood spot metrics for the analysis of peroxyredoxin 2 compared to whole blood samples. Spots were analyzed through Western immunoblotting assay within 24 hours from the spot. The results of our study on BAM3 and glycophorin A through flow cytometry demonstrated that both proteins decrease over storage time. In fact, they are progressively less expressed in blood samples stored for 20 days, indicated as T1, and 40 days, indicated as T2, compared to freshly collected samples, T0. This result is clear also in the protein ratio and suggests that storage affects erythrocytes volume and, consequently, the expression of BAM3 and glycophorin A. The experiment concerning BAM3 and peroxyredoxin 2 expression level through Western immunoblotting assay demonstrated that levels of peroxyredoxin 2 are significantly higher in blood samples after 40 days of storage, indicated as T2, compared to freshly collected ones, T0. On the contrary, BAM3 levels show a trend of decrease, but there is no significance between stored and freshly collected blood samples. This result can be explained with phenomena of accumulation of peroxyredoxin 2 due to increased oxidative stress. Moreover, the analysis of BAM3 and peroxyredoxin 2 levels in red blood cells fraction, consistently with previous results, show that peroxyredoxin 2 levels are higher in aged red blood cells compared to younger ones, while BAM3 levels are lower. These data can be explained with the abundance of aged cell in stored blood samples, since the renewal process is impossible in stored blood. Finally, peroxyredoxin 2 levels are measurable both from dried blood spots and from whole blood matrix. Levels of peroxyredoxin 2 of the same donor are slightly lower in dried blood spot matrix than in whole blood. This demonstrated that red blood spot can be a suitable matrix for the analysis of peroxide redoxin 2, but with less recovery than whole blood matrix. To conclude, we can say that BAM3 levels decrease progressively due to degradation that occurred during storage, while peroxide redoxin 2 increases. Consistently with this result, peroxide redoxin 2 levels are significantly higher in aged erythrocytes. BAM3, glycophorin A, and peroxide redoxin 2 can be considered promising biomarkers for storage to develop a direct method for the detection of autologous blood transfusions. The analysis of the alteration of BAM3 peroxide redoxin 2 ratio rather than the changes in the expression of only one protein can strongly improve the detection of stored and aged red blood cells. The aged red blood cells fraction may play a key role for the detection of transfused blood samples. Despite the impossibility to fractionate red blood cell subpopulation, dry blood spot is a suitable matrix for peroxide redoxin 2 analysis, but with less recovery than whole blood samples. Thank you for your kind attention.